Hi, I'm Ashley Marvin. We can understand brain activity using modern neuroscience approaches. I will go over two approaches today. The first approach is EEG, which we have discussed in class, and the second approach is calcium imaging. Both of these approaches are based on neural activity, so a knowledge of neurons is required in order to understand these techniques. Neurons. Neurons are nerve cells. They are the primary functional unit of the nervous system. The human brain has around 86 billion neurons. The neurons can be separated into four zones. The first zone is the input zone. This is where information is received from other cells. This information is received at the dendrites. The second zone is the integration zone. This is where it is decided if information is significant enough to be passed down, which happens at the axon hillock. The third zone is the conduction zone, where information is passed down. Information is relayed through the axon. Myelin sheath help relay information faster as it allows electricity to jump through each of the nodes of Ranvier. The last zone is the output zone, where chemicals are relayed at the axon terminal, which are then sent to the postsynaptic neuron. Membrane potential. Membrane potential is the difference in electrical charge across the neuronal membrane. The electrical charge is made up of the grouping of ions on the inside and the outside of the membrane. There are positively charged sodium ions, negatively charged chloride ions, positively charged potassium ions, and some other negatively charged ions that are referred to as organic anions. When a neuron is at rest, the sodium and chloride ions are more prevalent outside of the cell, and the potassium and organic ions are more present inside the cell. So at rest, the inside of the neuron is more negatively charged than the outside. The resting membrane potential of an average neuron is around negative 65 membrane voltage. Ions move across neuronal membranes by diffusion, which is differences in chemical concentration, or electricity, which is differences in charge. Ion channels are transmembrane proteins that allow ions to pass through. They are specific and only allow for one or a few types of ions to pass through. Ion pumps are proteins that actively transport ions. They move ions against their concentration gradients and require ATP. The sodium-potassium pump constantly pumps 3 sodium ions out of the cell while pumping 2 potassium ions into the cell. This helps keep the membrane potential negative. Calcium pumps transport calcium out of the cell. Potassium moves easily throughout the membrane through ion channels. Potassium will exit the neuron. Okay, so these pumps and channels are how these ions actually move throughout the cell. And so the voltage of the membrane would eventually change as like some positive ions are exiting the cell, negatives are entering, and different ions keep moving around. And then we'll discuss what will cause that now. Potassium will exit the neuron until it reaches a point where it's at equilibrium, which is when forces like diffusion aren't pushing it in one direction or the other. When neurotransmitters bind to the receptors on the dendrite of the neuron, they can cause depolarization of the neuron. So neurotransmitters, like I said earlier, are released from the presynaptic neuron to the postsynaptic neuron, and the dendrites are what at the top, at the input zone. So when these neurotransmitters bind to receptors on those dendrites, that's when depolarization of the neuron of the postsynaptic, that's when depolarization of the postsynaptic neuron will happen. All right. Depolarization is when the membrane potential becomes less polarized or closer to zero, so it'll become less negative. Once the membrane becomes depolarized enough to reach its threshold potential, a large number of sodium channels will open, allowing positively charged ions into the cell. This causes massive depolarization of the neuron as the membrane potential reaches zero and becomes more positive. The influx of positive ions creates the electrical signal which travels down the neuron. So that same electrical signal that I was talking about earlier, which is the information that's passed down from one neuron onto the next. The action potential will reach its peak and sodium channels will close and potassium channels open, which allows potassium to flow out of the cell. The neuron would repolarize as it loses positive potassium ions, so it's becoming negative again. This is the falling phase. The neuron returns to the resting membrane potential, but then it passes it and overshoots it, so that it's even more negative than the resting potential, or hyperpolarized. This is the refractory phase, in which it is very difficult to fire another action potential. The potassium channels will then close and the membrane will return to resting membrane potential, in which another action potential can be fired. An action potential travels down the neuron and causes the release of neurotransmitters at the axon terminal to pass on to the postsynaptic neuron. So that's how information is relayed in our body. That's how we receive signals from external stimuli, and then use it to relay information to our brain so that we can respond to these stimuli. So when we're looking for, when we're recording brain activity, we often look for electrical activity through neurons, because that's how we're understanding our environment and responding to stimuli. And we will not be talking about glial cells today. Okay. As discussed in class, one approach in measuring activity in the brain is EEG. EEG measures electrical activity in the brain, which is produced by neurons, as just discussed. Electrodes are placed on the scalp, and they pick up electrical signals generated in the brain and transmit them to a machine that records them as the EEG. EEG does not record the activity of single neurons, but rather detects the signals created when populations of neurons are active at the same time. So if you see like little waves for the EEG graphs, like on the right, those are recordings of basically how many action potentials are occurring at the same time, like in groups. So none of that is like the recording of a single action potential. And also action potentials all have the same amplitude, so that might be easily mixed up, but if it was a single action potential, you're like, oh, it's how much it's spiking. No, they're all the same. And this records how many action potentials are going off at the same time. Okay. EEG mostly records signals from small areas of the brain surrounding each of the electrodes. EEG is primarily measuring postsynaptic potentials. So postsynaptic potential, again, is after the second neuron receives the information and then fires an action potential. So either measuring postsynaptic potentials or changes in membrane potentials that are elicited by neurotransmitters binding to receptors on the postsynaptic membrane. EEG provides an image of electrical activity in the brain represented of waves of varying frequency, amplitude, and shape. A small amplitude indicates that neurons are less activated and are firing asynchronously. So a shorter wave would mean less neurons are activated and they're not firing at the same time. And then a larger wave would be the opposite. A larger amplitude would be the opposite. It indicates that many neurons are being activated at the same time. EEG can be used to measure brain activity that occurs during an event, or to measure spontaneous brain activity that happens in the absence of a specific event. The typical electrical activity that occurs in association with an event is sometimes referred to as event-related potential. EEG is commonly used to diagnose epilepsy and characterize seizure activity to monitor sleep for the diagnosis of sleep disorders. So for epilepsy, for an example, it's often seen, or epilepsy is often characterized as an abundance of action potentials firing at the same time, like just way over the normal. So in an epileptic brain, while there's an epileptic episode, the amplitude would be extremely high. And then it is also used to monitor sleep for the diagnosis of sleep disorders in an effort to diagnose or provide information about a variety of other types of brain dysfunction. While there are many advantages to using EEG, such as its low cost and ability to measure brain activity on the order of milliseconds, there are some other limitations as well. Because EEG monitors activity in large groups of neurons, it is difficult to pinpoint activity using EEG to a precise location in the brain. And it records the activity of neurons in the cortex, but is limited in its ability to accurately record activity in deeper structures of the brain. So my second method is actually able to record a lot of individual neurons so that you can specify locations in the brain and you can specify what these individual neurons do. Okay, so here's our next method, calcium imaging. Calcium imaging measures neuronal activity by adding a chemical dye or genetically encoded indicator that changes fluorescence in response to calcium. The brain is then imaged with a microscope. Calcium imaging allows activity in many individual neurons to be measured, as increases of calcium are associated with neurotransmission. Calcium imaging can be used to locate areas in the brain that respond to specific stimuli. An example of this is the research of the gustotopic map of taste qualities in the brain. So that would be looking for like which areas in your brain respond to specific tastes. This research used calcium imaging on mammals to examine neural activity in response to taste stimulus across the gustatory cortex to understand taste mapping in the brain. First, they confirmed the gustatory area in the thalamus by using extracellular electrodes. A sweet stimulus was then used to identify taste-responsive neurons. So they basically use this chemical so that when there's an increase in calcium, the chemical will change the color of the neuron. So they know when the neuron's being activated because of that increase of calcium, because it's now showing a different color. And so they can see which neurons are being activated after they use specific stimuli. So this doesn't just have to be used for taste. This is just one specific example. Okay. So a sweet stimulus was used to identify taste-responsive neurons. Cells around this area were infected with an AAV2 whole GFP virus to appear green, and then these are the dyes that they used. They stay in the coronal section with the 2-Pro3 to appear red, and the intersection between the RV and the MCA was injected with DIL to appear blue. The second layer of the gustatory cortex was inspected for taste responses and neurons showing patterns of taste-responsive firing. A stimulus paradigm of artificial saliva and exposure to a test agent and a post-stimulus wash were used to trigger and analyze responses in the brain. This was done for each of the 5 tastes: bitter, sweet, umami, salty, and sour. Through these experiments, researchers were able to identify that the neuronal taste responses for a specific taste are spatially separated. These locations are distinct hotspots in which many neurons respond to taste stimuli. The calcium imaging made these hotspots evidence as the taste-respondent neurons emit fluorescence when active. Once hotspots were located, different tastes were tested to see if a specific taste hotspot would activate for another taste, demonstrating that each hotspot only activates for their specific taste. To further test the particularity of the taste-responsive neurons, the researchers used a specific bitter taste stimulus, cyclohexamide, on knockout animals who were without the T2R5 receptor. Knockout animals, they're genetically modified so that they lack like a specific receptor in this case. They're using the stimulus cyclohexamide on a rat that doesn't have the receptor that responds to cyclohexamide, and then on a normal one. And then this resulted in no responses to cyclohexamide in the knockout animal as expected, but that knockout animal still responded to other bitter stimulus, which just shows how specific it just shows how specific like the receptors and the neurotransmitters are for specific stimuli. And this one's just specific for taste. Okay. Similarly, respective tests were performed for sweet taste, which had the same outcome. Through this research, we have a better understanding of the taste system. It provides a distinction of taste respondent cells to their specific tastes and a gustotopic map, which demonstrates the separation of taste hotspots in the insula. And these are just two examples of modern neuroscience approaches that help us understand how our brain functions, and like we can really just target which areas of the brain correspond to specific functions. And these are just two of them, and there's so many. Neuroscience research methods help us understand how the brain functions and prove to be extremely useful in understanding different brain regions and their functions. So that's all for today. Thank you. Oh my God, where'd it go?